students, how are you all? So students, today we will discuss about one chapter is there in 12th class, that is known as the biotechnology. Okay, so we will discuss that biotechnology chapter thoroughly. First of all, let me to say you some introduction. Okay, before going to inside the chapter, first of all we have understand that the meaning of biotechnology, what is the meaning of bio and what is the meaning of technology altogether. Okay, so let us discuss biotechnology. Listen, bio means what? Especially the meaning bio means what? Bio means life science. Yes, bio means life science. And what is the meaning of technology? Technology means one technique. Understood or not? You can say it by process. Okay, technology, techniques, processes. Yes or no? Let me to write here. Processes or techniques. Okay, so many techniques are there, so many processes are there. In life science, whenever such processes, such techniques, it will be introduced. Yes or no? Introduction of some process, of some techniques, whenever it will be introduced inside the life science, yes or no, inside biology, that is known as the biotechnology. And how it will be done and why it will be done, why it is necessary to introduce your technique, any kind of process, I will say you. Obviously the point is that for the benefits of your human society, for the benefits of society, yes or no, to get the advantage of something what we have lacking in some processes. Suppose we will feel that something is needed there due to the lack of such kinds of substance, any kind of organism, yeah, the population of any kind of organism they're suffering from. Okay, the point is that whenever any other kind of item will be introduced in their body, then they will not suffer. Yes or no? This is the point. So let me to discuss with you in brief manner. Understood or not? The point is that biotechnology, whatever I told you, that introduction of some process inside the life science, that is known as the biotechnology. So what is it mean? Suppose we have the one point that is known as the gene. Everybody, you might have listened that word gene. Yes or no? Gene is the what? Gene is one coding segment of DNA. DNA means deoxyribonucleic acid. It is present inside nucleus. Understood or not? So gene is what? Gene is the coding segment of DNA. Understood or not? For our each and every characters, gene is responsible. Who is responsible? Gene is responsible. So different kinds of codings are there present in DNA. Yes or no? One coding segment, that is called the gene. Understood or not? The point is that here let me to give you another introduction, another information, that is what? Another word you might have listened, that is known as the genetic engineering. Genetic engineering. Yes or no? Genetic engineering is one part of this. One is genetic engineering, another is chemical engineering. Yes or no? Another word is that is known as the recombinant DNA technology. RDT. Recombinant DNA technology. Yes or no? The point is that. Here let me to say again that biotechnology means, right now also you might have been so many doubts. Yes or no? Listen, suppose this is one organism. Okay, and this is another organism. Okay, roughly let me to say you that this is one organism, this is another organism. Suppose this organism having some problem. Yes or no? Due to lack of one gene, yeah, defective gene, yeah, any genetic disorder. Due to any genetic disorder, due to any defective gene, yeah, due to any kinds of gene absence in this organism. Organism number one. In this organism, due to any genetic disorder, this organism is suffering from any kind of disease. Yes or no? So the point is that we have to introduce that type of gene inside its body, so that in future this body will not be suffered from such kind of diseases. So the point is that we will search that from where we will bring that kind of gene, so that we will introduce in the body of this. Suppose we will search anywhere else, so many places we will just search, and after searching so many things, suppose we will get that in organism 2, another organism 2, suppose this organism does have that kind of gene, yes or no? That gene is the suitable gene, what we can transplant, what we can transform here, we can shed here, so that this organism will not be suffered from that kind of disease anymore in the future. Yeah, its next population. Understood or not? So whenever we will remove that gene from this organism, obviously we will cut DNA. Gene is present here inside the DNA. So we will cut that some fragment of DNA. Suppose inside it one fragment of DNA is present. Suppose this is the DNA. Okay. We will cut some fragment of DNA where that gene is present. Gene is the coding segment of DNA. So all over that DNA will not need that. Only some specific point we will cut. Specific point we will cut. What DNA holds particularly that desirable character. What desirable character is lack in this organism. That desirable character is needed in this case. And what desirable character is not present here, yeah, due to any kind of problem, genetic disorder is not present here in lacking portion. So wherever that gene is present, where that desirable character is present, yes or no, that coding segment we will remove that one. That portion, small segment of DNA we will remove from there by different types of tools. So many tools are there, yes or no? Different types of enzymes are there. They help us for such kinds of things, yes or no? So what are tools are there? We will discuss. Just like that, restriction endonuclease enzyme. Okay, restriction exonuclease enzyme. So different types of enzymes are there present. Okay, we will remove that one and then we will set up inside this organism. And for setting up inside this organism also one specific enzyme also helps us. So the point is that alteration of genetic materials. Alteration of genetic materials means genetic engineering means what? Whenever genetic materials will be altered from one organism to another organism for benefit purpose. Yes or no? So that any of the organism will get that advantage. Yes or no? Engineering means manipulation. Engineering means what? Manipulation. Whenever manipulation or alteration will be done, whenever transformation of genetic materials will be done, whenever carrying away of genetic materials will be done from one particular foreign material or foreign organism, whenever that desirable character having that gene will be transformed into, okay, the organism having any kind of genetical disorder, whenever will be transformed from there to here, yes or no? And that is called the recombinant DNA technology. Recombinant DNA technology means whenever recombine means what? Whatever DNA small fragment will be caught here, that small DNA fragment whenever it will be added there. Previously one specific DNA is present there, but the point is that what small fragment it will be cut down. That small fragment whenever it will be added here, so that is known as the recombine. And recombine of your what? DNA. Yes or no? And this DNA recombination, it was done there. Yeah, it will be done there with the help of some tools. Yes or no? And that is one technique, and that is known as one technology. So this is known as the recombinant DNA technology. Yes or no? The point is this. Understood or not? One interesting story is there behind it. Yes or no? What happened? You might have listened everyone one word that is known as the insulin. Yes or no? Insulin. Okay. One word is there that is called the insulin. Insulin is secreted by pancreas. Yes or no? Insulin, this is hormone, but it's secreted inside from pancreas. Yes or no? Whatever present just below the stomach region. The point is that insulin is very lack in case of some specific patients, those who are suffering from diabetes. Diabetic patients, yes or no, that may be diabetic insipidus, diabetic mellitus, yes or no, such kinds of patients, they are having lack in their body insulin. That's why insulin injection is there given to them to manage that ratio of insulin inside the body. Yes or no? So what happened? Previously this insulin, this hormone, it was extracted from one animal that is known as a pig. But right now insulin, that hormone is not extracted from pig. Okay, this is not a related issue. So pig is the animal. Previously it was extracted insulin from pig. So the point is that from the pancreas portion of pig, whenever insulin was extracted from pig, so what happened here? and it will be installed inside one human being's case. Okay, so that it was seen there that some infections are there happening after the installing, after giving the dose through vaccination process. Whenever peaks insulin it was then installed inside human beings case, so some serious allergy condition, some infections were there carried down. Yes or no? So at that point what happened? Listen, inside our body actually one system is present that is known as the immune system, immunoglobulin system. Yes or no? Yeah, what you say that immunity system. Yes or no? So due to that system, our body whenever recognizes any kind of foreign materials that coming inside our body, suppose any kind of foreign protein coming inside our body, whenever our body it will respond that, suppose our body will be recognized that yes, this is the foreign material, this foreign material coming out inside the body. So our body will our body does have one kind of system that is known as the immunity system, by what our body will produce antibodies so that foreign materials, foreign proteins will be thrown outside. It will be killed there, it will be engulfed there, and it will be thrown outside. Yes or no? It will be declared as molecules and it will be thrown outside. So that is the point. Okay, so here what happened? Allergic reactions it will be happen. So if this insulin it will be given to body, and our body due to going up different types of inflammations, allergic reactions, our body whenever will generated antibodies, then antibodies will kill that antigens. So in the next time insulin it will not be acted. So the point is that whenever we will give the insulin upon human being's insulin, yes or no? Whenever insulin will be extracted from one particular human being, where insulin will be found, actually insulin is produced from inside everyone's body, yes or no, from pancreas. Whenever human being's insulin, whenever it will be extracted, and that insulin whenever it will be introduced inside the body where insulin is lacked, then it will not be caused any kind of allergic reaction. So that is the point. Here shortly I am just giving a rough diagram that suppose this is known as the bacteria. Yes or no? This is known as the bacteria, and this bacteria is known as the Escherichia coli. Yes or no? Escherichia coli. And this is known as the DNA segment of any human body. Yes or no? And this is the DNA segment, and this is known as one coding segment of this DNA, which is called gene. Okay, obviously it will be seen like this. Yes or no? This gene will be seen like this, just like a DNA. Okay, so the point is that whenever we will remove this one, understood or not? This gene whenever it will be removed, another thing is that this DNA does have one kind of property that to replicate itself. It can replicate itself. Replicate means it can give some identical types of itself. Okay, the point is that through some central dogma, it can transcribe itself to form messenger RNA. Understood or not? Through central dogma, this is the process. Okay, this is the process, nothing else. We will discuss thoroughly that what is this central dogma and transcription. Okay, translation. So through central dogma, this DNA, okay, it will be formed, it will be transcribed into messenger RNA. Yes or no? Then after that kind of protein it will be formed, what can be functionable. Yes or no? And this is known as the insulin. Understood or no? Because one coding system is present there between behind each and every genes that to perform some specific type of tasks. Yes or no? Different kinds of genes are there present. Some genes are responsible for height. Some genes are responsible for producing melanin pigments. Some genes are responsible for doing different kinds of things. Similarly, this gene is producing insulin. Okay, because coding is there. This gene is responsible for insulin only, and it will be insulin. It will be produced that. Understood or not? Whenever this gene it will be removed there and it will be installed inside this in bacteria Escherichia coli. Where it will be installed? In Escherichia coli. Yes or no? How in detail I will discuss with you. Okay. Then in this bacteria already in previous manner this bacteria did have its own DNA. Whenever this gene it was removed from here, and that small segment whenever it was added there. So this is known as the recombinant DNA. Yes or no? Because that DNA consists of that gene. Yes or no? The human beings, that gene, what is responsible for producing insulin. So next, whenever this bacteria will do its reproduction and so many clones It means different types of bacteria will be produced through replication process. All such bacteria will have that certain type of characteristic. Okay. Then after that it will be purified, it will be isolated, and then it can be produced vaccine. It can be launched in the market. So then after that vaccine can be given to that person where insulin is lack. So this is the point. This is the shortly I was saying you about the some idea. Okay, I was discussing with you. Actually, in the next class, in the next page, in lecture number 2, I will discuss with you the proper definition of biotechnology, and thoroughly we will discuss that in the next steps. Okay, thank you.